Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. Now, it is the time for lecture 42, where I will talk about the biosynthesis of carotenoids and carotenoid cleavage products. Let us go to the board. So, here we will talk carotenoid biosynthesis and carotenoid cleavage products. So, now go to the board. So, carotenoids. What are carotenoids? So, carotenoids okay, basically the C40 compounds tetraterpenes, but I will introduce this from the point of distinctive color. So, carotenoids have distinctive color. So, the color such as which ranges from yellow to red spectrum. And this is due to a series of conjugated double bonds. This is due to the presence of a series of conjugated double bonds. I will draw the structure of beta carotenes a little bit later. And diverse modification to the simple polyene chain structure. So, the carotenoids contain the polyene chain structure POLYENE, polyene chain structure, and where modifications occurred, and as a result of this modification there will be changes in the color range. For example, astaxanthin. Astaxanthin are usually red in color. But this astaxanthin, when it is present with the crustacyanin protein, which is present in the shellfish, for example, lobster. So, that results in blue pigmentation. And then what happens, that when this is cooked, this turns to red again, because during the process the protein got degraded. That means, the carotene compounds can linked with a protein leading to a different color, and then when the protein got degraded, then it brings back to the original red color, and that is why you see that the lobsters, when they are cooked, the shell turns to red in color. Okay. So, carotenoid backbone, what about the carotenoid backbone? Let us see. So, the carotenoid backbone is either a linear or it contains cyclic group. And when it is the cyclic group, the most abundant end group is a beta ion ring. And the example of this is beta carotene. And the carotenoids containing this beta ion actually act as important precursor for vitamin A. So, other cyclic end groups are eta ring. So, which is commonly found in plants. Along with that, there are unusual cyclopentane ring. Unusual and the examples are capsanthin, capsanthin, capsanthin and capsorubin. So, what are these? These basically imparts red color to the paper, red paper. So, the red color comes from this, but these are unusual carotenoids and it contains the cyclopentane ring. So, based on this, what are the two major classes of carotenoids? Two major classes of carotenoids. One is the carotene and another is the carotene with oxygenated derivatives, which are basically called the xanthophyll. And the examples are lutein, violaxanthin, and which are the key components of LHC complex, and normal carotenes are there. So, this is about the brief, and now what I will show? I will show you some structure in the next slide. protein chair. So, this 4. So, this is basically the structure of lycopene. Most of you heard about the lycopene, which is the principal pigment in red tomato, and now I will draw the structure of beta carotene. So, this is the structure of beta carotene. Now, you will also come across with another name, which is called alpha carotene. So, alpha carotene, I will not draw the whole structure, only a part, that is so, here is the difference, that is here, where is in beta carotene, the bond is here. Okay. And what I said, that the beta ion. So, this is basically the beta ion ring. So, what happens, that lycopene undergoes cyclization. So, there are two cyclization, one is in the left side, one is in the right side. So, that you must appreciate. So, the beta ion, if I draw singly, then it should be like this, that so, beta ion is cleaved out and it is itself a part of the volatile compounds. Okay. Now, let us go to the biosynthesis of this carotene pathway. Okay, the next slide. Carotenoids. So, we start with GGPP, which we have studied before, geranyl pyrophosphate plus geranyl pyrophosphate. It two joins together by the action of an enzyme called phytoene synthase. PSY stands for phytoene synthase, and which makes phytoin. Phytoin is colorless. This phytoin will be converted subsequently into lycopene. Now, if we dissect this, we will study the pathway further, but now what I am doing here is basically I will try to give you some more information about these early steps. The phytoin, what is formed here, it is called 15 cis phytoin, and this will be converted into 15 cis phytofluin. That will be converted to all trans phytofluin, and that subsequently it will be converted into zeta carotene. And so, and if we put the pathway in this side, so the phytoin will be converted into zeta carotene, and zeta carotene will be converted into tetracis lycopene. Trans lycopene or trans, just a moment. Zeta carotene, tetra cis lycopene, and okay, let me use the red color. And that will be converted into all trans lycopene. All trans lycopene. Zeta carotene okay, so here so zeta carotene will be converted into any neurosporin. Now, So, the enzyme, this is PDS, this is ZDS. PDS stands for phytoene desaturase. ZDS is this zeta carotene desaturase, and then the enzyme is CRT iso, CRT iso, CRT ISO. CRT ISO stands for carotenoid isomerase. And this all trans lycopene will now be converted into gamma carotene. Sorry, it has to be in that color. So, the enzyme here is beta cyclase, beta LCY. Beta LCY stands for beta lycopene cyclase. Okay. Now, this can follow two paths. The one path which makes beta carotene. So, the enzyme is here beta LCY, beta LCY, and the other one which makes alpha carotene. So, that enzyme is eta LCY. This makes alpha carotene. And this alpha carotene subsequently converted into xenoxanthin, ZEINO, ZEINO, XANTHIN, and this will be converted into lutein. Lutein is yellow color. So, but just to distinguish, if too much lutein is accumulating, then it turn into red color. So, to distinguish it from the beta carotene, I put it a different color here, and here the enzyme is, first one is a beta hydroxylase and the second one is eta hydroxylase. Okay. Now, from beta carotene, the pathway contributes to beta cryptoxanthin, and that subsequently makes zeaxanthin. And zeaxanthin, okay, because of the shortage of space, I think from gamma carotene onwards, I will show it in the next slide. So, the enzyme responsible for these steps up to beta cryptoxanthin is again a beta hydroxylase. So, let us go to the next slide. So, starting from this is about some apocarotenoids. So, but I go here. Gamma carotene makes the left side alpha carotene, xanthine, and then finally lutein. Okay. So, this is eta LCY beta hydroxylase, and this gamma carotene is coming from all trans lycopene. I will also show that point of all trans lycopene. And the enzyme is beta cyclase, beta lycopene cyclase, beta LCY. Now, this side all trans lycopene converted into beta carotene. Now, all trans lycopene converted into gamma carotene, and this gamma carotene will be converted. What is the pen color? Converted into beta carotene. So, this beta carotene will be converted into beta cryptoxanthin, sorry, always to xanthin. This will converted into zeaxanthin. This will be converted into antheraxanthin. This further into violaxanthin. Here is a different color. Violaxanthin, and violaxanthin will make neoxanthin, and let me join it first. The enzymes beta carotene to beta cryptoxanthin is beta hydroxylase. This is beta LCY. Okay, then this one is again another beta hydroxylase. Then this ZD stands for zeaxanthin epoxidase. Again, this will be another zeaxanthin epoxidase. and this will be neoxanthin synthase, NXS neoxanthin synthase. Okay. Now, from this neoxanthin and violaxanthin, maybe nine cis neoxanthin 9-zin violaxanthin can be formed. And which contributes to the formation of stress phytohormone abscisic acid. Okay. Now, two things to be noted here, that I have shown you the structure of alpha carotene and beta carotene. So, it is similar, only the beta ion ring in the left side, the double bond position is different. So, you see here that from all trans lycopene, in order to formation of alpha carotene, there will be two cyclization. First one will be catalyzed by beta cyclase and the second one will be catalyzed by the eta cyclase. Whereas, for the beta carotene formation, again here will be there is a two cyclase cyclization. First one will be catalyzed by beta cyclase and second one is also catalyzed by beta cyclase. This is the difference, and which there is a hydroxylation. So, essentially the zeaxanthin and beta carotene structures are similar, only that at two position two hydroxy group is added, and that is why the OH and OH. So, first OH gives one OH addition in one beta ion ring, the other OH gives the another hydroxyl addition in the other beta ion ring, and it makes the zeaxanthin molecule. So, these are important carotenoids and these are the starting point for different carotenoid cleavage products. So, what I can do now, I can briefly talk about the carotenoid cleavage products. So, carotenoid cleavage products I have already drawn for you in one of the previous slides, which I can show you. Okay. So, okay, yeah, I have drawn it for you. So, carotenoid cleavage products, beta carotene will be cleaved and the carotenoid cleavage products are called apocarotenoids. So, CCD stands for carotenoid cleavage dioxygenase. CCD stands for carotenoid cleavage dioxygenase. Okay. So, as a result of this cleavage, you see that beta ion is cleaved out. So, that I said itself is a volatile compounds. The other portion of the cleavage can generate another compound, which is called dialdehyde. Similarly, beta cyclocitral is also formed as a part of the cleavage, and the other carotenoid cleavage product is beta apo 10 carotenol, which undergoes further cleavage leading to the formation of another smaller molecules. So, that we will see here. Beta carotene, the carotenoid cleavage dioxygenase 7 cleaves beta carotene into beta apo 10 carotenol, which is C27, and beta ion, which is the beta ion. And in the next step, what happens? This beta apo 10 carotenol undergoes further cleavage by another carotenoid cleavage dioxygenase 8, leading to the formation of beta apo 13 carotenone and dialdehyde, which I have also shown here. So, beta apophthalate and dialdehyde I have shown here. Okay. And this is the structure of zeaxanthin. As I have said, it is similar to beta carotene, but one hydroxyl group here, one hydroxyl group here, and this undergoes cleavage by the enzyme called zeaxanthin cleavage dioxygenase, that leads to the formation of quercetin glucosides along with another molecule called safranal. And similarly, from lycopene, the another carotenoid cleavage molecule formed, which is called bixin. So, bixin basically is found in the plant called Bixa orellana. So, Bixa orellana seeds the seed coat, which is resinaceous in structure. So, that basically the place where bixin accumulates. So, this is the structure of bixin here, and bixin is basically used for coloring of the food ingredients, including the Amul butter what we consume, that yellow color comes from the bixin. Now, zeaxanthin I just briefly tell in one of the next slides. For example, the zeaxanthin undergoes again this cleavage by the enzyme called zeaxanthin cleavage dioxygenase, ZCD. I am not writing the full form, it is pretty clear. Zeaxanthin cleavage dioxygenase, leading to the formation of a compound called quercetin dialdehyde. Quercetin dialdehyde subsequently got converted into quercetin by the action of an enzyme called aldehyde oxidoreductase. And quercetin further converted into quercetin glucoside, which is called crocin. So, by the enzyme called UDP glucosyl or glycosyl transferase. Okay. So, I have shown you the structure of this zeaxanthin. I have shown you the structure of beta carotene. As I have said, it is pretty similar, only that this one hydroxy is here, and I will not draw the whole thing, it will take a long time. Okay, double bond here, and finally, when we see the structure of quercetin glucoside, because it is cleaved. So, it is like this is the glucose moiety attached. So, this is the quercetin glucoside. So, that means the quercetin, when you see the quercetin structure, this is HCOC, and when you see the crocetin aldehyde, then basically this is this CHO. So, this is how this transformation happens, leading to the formation of very important and acceptable food colorant, which is this crocin, and this is stored in the vacuole. So, which makes slightly this is as water soluble as well. So, with this there is only a small portion left. I have mentioned about the Bixin. So, I just take only another 1 or 2 minutes for lycopene. Lycopene makes Bixin aldehyde. Lycopene makes Bixin aldehyde. So, Bixin dialdehyde, and that forms the Norbixin, and from there Bixin is formed. So, you see in the Bixa aureliana both Bixin and Norbixin are present, and here there is an formation of this methyl transferase enzyme which converts, and here there is a dehydrogenase, and this is the lycopene cleavage dioxygenase. So, this is in brief about the formation of Bixin and formation of crocin. So, in this class I have spoken on the biosynthesis of carotenoids and also the different carotenoid cleavage products, which are very important molecules or having lot of practical applications. So, with this I end this class. Thank you.